Great. Thanks a lot, Julian. Thanks for the opportunity to present today. We do quite a few things on Aptamers in our lab, but today I'll be presenting some work on thermostable light-up fluorescent Aptamer. So just a little bit of background about fluorescent light-up RNA Aptamers. So generally they bind to a fluorogen, which has no fluorescence. When the Aptamer binds, it actually elicits a high fluorescent signal. And there are many examples already in the literature, spinach, broccoli, mango, corn, robust. I've got some figures here from the original spinach paper. And so you can see the fluorogen on its own has no fluorescence. Obviously, the RNA on its own has no fluorescence. But when there's the RNA and the fluorogen together, they have a very high fluorescent signal. And that's very specific for that individual RNA aptamer. And so this can be used for tracking RNA in a cell. So generally, these aptamers are selected using binding capture selection. But that is really just for selecting for binding affinity. It doesn't actually select for fluorescence activity. So more recently, we can use microfluidic droplet selection to select actually for a turn on in the fluorescent signal. So this has three main steps. There's droplet generation, pico injection, and then droplet sorting. So droplet generation, for this, basically, it's a water in oil emulsion, which is generated on a chip. And you can see here the aqueous phase coming in from the left and the oil phase coming in from the top and bottom to form a sheath and make the droplets. And so in these droplets, there'll be PCR reaction mix, and then also the template of interest, which will be in a Poisson distribution. So there'll be like 0.2 template molecules per droplet. And then that ensures that you have a monoclonal amplification in the droplets. So after this droplet generation, you go off chip and go to a thermal cycler. After the PCR, you then inject these droplets back onto the pico injection chip. And so you can see them coming in from the left here. And then in the middle, there's actually a pico injection of transcription mix, and then also the fluorogen of interest. So after this, the droplets go off chip for another incubation, this time 37 degrees Celsius. And this is for the transcription to occur. So, following transcription, then you've actually got the active RNA aptamers in the droplets with the fluorogen. So then you actually have a fluorescent signature on each one of the droplets. And so you can do this under a microscope or inject them into the droplet sorting chip. And then, the droplet sorting chip basically has a laser and then also a filter and a light sensor. So you can detect the fluorescence of every individual droplet. And then if it's a high enough fluorescence over the threshold, then you can sort into the positive channel. So this is using a dielectrophoresis effect of the two electrodes here. So following this droplet sorting, then you just recover the DNA for the next round of microfluidic selection or actually for sequencing. Okay, here I'm just a movie of the droplet sequence sorting happening. So you can see the positive droplets in the dark color being deviated into the positive channel. And so this is slowed down actually. The sort rate's around about 250 per second. And so, this approach has been used previously for selecting RNA light-up aptamers. So we've got iSpinach and also mango 3. And so, whenever you're going to do an in vitro selection, you're not really guaranteed that your aptamers are going to work well inside a cell. And so it's very important to take this into account because if the downstream application is, tracking RNA in a cell, they need to work very well in a cell. So, with this in mind, we tried to make a intracellular mimic for the microdroplet environment. So there's many molecular interactions occur in a cell. So some are molecular diffusion, charge interactions, hydrogen bonding, solvent polarity, and base stacking. So you could take cell-free media and insert this into these droplets, but that detracts a little bit from the functionality of these droplets. You can merge them and split them and do all kinds of interactions with them. So we wanted like a very simple way of mimicking the intracellular complexities to have a selection for these. So we chose temperature. Obviously, this has been done before previously. The other thing we chose is low salt concentration. So, you know, that sounds a bit strange because if you've done selects before, usually you want to increase your salt concentration to get very specific aptamers, get rid of the non-specific binding. But actually, the salt helps an aptamer fold. So the aptamer has a negatively charged backbone, and so it needs the positive charges to be able to fold into that conformation. And so if you ever use aptamers in deionized water, they just won't work. So this is actually a very good selection pressure, is having very low salt to then select aptamers that fold very well in a difficult environment. So for the starting material for these droplet-based selections, we used the broccoli aptamer attached here to a tRNA, and then we performed mutagenic PCR to make the library. And so yeah, just going over the microfluidic droplet selection again, it's droplet generation, PCR, pico-injection, transcription, droplet sorting, and then recovery of that DNA. And so we did 5 selection rounds here. And so you can see for the selection temperature, we started off at room temperature, and then we went to 37 degrees Celsius and finally 42 degrees Celsius. For the salt concentration, we kept it at 5 millimolar. And for the error-prone PCR or mutagenic PCR, we did the 0th round, the initial library, and then the 2nd and 4th round. And so in terms of the, some data from the selection. So this is just some very typical data. So here you can see a microscope image of the droplets. This is after transcription, and you can see they have some droplets with a high fluorescence, others with a low fluorescence, and some in between. And a histogram of that data, you can see we've got some, high fluorescent droplets, but many low fluorescent droplets. So we usually have a threshold around here to just select the highest fluorescent droplets. And, following NGS, we got a lot of data, and we found that there were mutational hotspots within our broccoli sequences. So, and you can see here the mutational hotspots on the left. They're not just regular base pairing regions, they're sections that are involved in the actual binding to the fluorogen. So yeah, looking at some of these variants in vitro just to analyze their performance. So this is just a simple plate reader assay with the fluorogen, the aptamer, and the buffer. And we measured at different temperatures, 25, 37, and 45 degrees Celsius. So on the left, on the far left, you can see the original aptamer. And in the increasing, with the increasing temperature, the fluorescence intensity actually decreases. Whereas our triple mutant on the end here, even at the high temperature, 45 degrees Celsius, we still have a very strong fluorescent signal, very similar to 25 degrees Celsius. And so this is our triple mutant here, which we termed enhanced broccoli or eBroccoli. And so see we have 2 mutations in the loop on the stem, and actually 1 next to the G quartet here. So yeah, next we wanted to look at this in cells. We started with E. coli. So for these experiments, we measured, so we cloned the aptamer sequences into a plasmid, also with mCherry as a normalizing control. And so for the top panel, we've actually got the original broccoli, and the bottom panel is enhanced broccoli. So you can see at the high temperature, 45 degrees Celsius, the original broccoli, the fluorescence intensity actually decreases for these cells, whereas for our enhanced broccoli, the fluorescence is very, very stable. So that's the same data expressed over here in a bar chart. And so at 37 degrees Celsius, which is really the most important temperature, we've got a 3.7-fold increase in the fluorescence intensity. And so just going into that data a little bit more, for our original broccoli sequence, at 37 degrees Celsius, we've got a lower fluorescence intensity. So that tells you that basically that aptamer is in equilibrium between being folded and unfolded. So if you think of that, if you're doing microscopy, that's your fluorescent signal kind of ebbing in and out of existence constantly. So it's not a very good situation for imaging. whereas our enhanced broccoli has a very strong fluorescent signal even at a higher temperature. So we've just shifted that equilibrium of folding. And so, yeah, next doing some mammalian work. Basically, we constructed in a plasmid beta-actin CTS UTR with 6 repeats of our eBroccoli 3-way junction and a poly A tail on the end. And transfecting this into HEC 293 cells. See, we've got the DAPI in blue, the RNA fluorescence in green, BI. And to the top, we've got the original broccoli, and the bottom, eBroccoli. And so you can see it's very similar to the bacterial work. It's a 3.9 times fluorescent enhancement with our new Aptamer sequence. So yeah, now we wanted some application. We really wanted to test this Aptamer and see if we could get it to work well for a certain application. So we chose stress granule formation via liquid-liquid phase separation. So, you know, for liquid-liquid phase separation, it's basically a network of interactions, very small molecular interactions, which then forms a cluster and can perform, you know, functions in cells. So protein-protein, RNA-protein, and RNA-RNA interactions form nanoclusters, and then through coarsening, we get stress granules. And again, looking, here are some cells that are transfected with our plasmids, and you can see the original broccoli. Initially for these time course experiments, the temperature is 37 degrees Celsius. After this point, the temperature is increased to 42 degrees Celsius to stress the cells and induce the formation of stress granules, and imaged over about 100 minutes. So for the original broccoli, you can see the fluorescent signal doesn't really last for this entire time, just disappears. But for our eBroccoli, the fluorescent signal can be seen throughout the whole time course. And you can see the data is plotted over here as well, showing that. And so here we've just zoomed into the eBroccoli data. And so you can see at 37 degrees Celsius, the RNA is, you know, localized in the cytosol as it should be, and it's more dispersed throughout the cytosol. When the temperature is increased to 42 degrees Celsius, you can see there's many more puncta emerging on the screen. And even at 100 minutes, you can see there's many more puncta on the screen. And so these are mobile within the cytosol, so it's actually got the same you know, function as the wild type, our tagged RNA. Yeah, so just looking at this work, actually the original broccoli wasn't selected in vitro, it was actually selected in vivo. It was already selected in bacteria and then selected using FACS. So it should actually perform in vivo really quite well because it was selected in vivo. But we've actually managed to make a better variant here using an in vitro technique and just modifying the selection parameters in this way to really select for folding in these like high stress cellular environments. And so, yeah, you know, microfluidic droplet selection is very useful. And this is just one application. You know, any assay that can be tied to fluorescence can be used for this approach. So it could be selecting for ribozymes or catalysis or something like this. So yeah, I'd just like to thank Julian Tanner for his help and support. Also Lin Wang, who did the microscopy that you've just seen now. And also our collaborators from engineering for their microfluidic help. And so that's Professor Anderson Shum and Gorway. Yeah, I'm happy to answer any questions you might have.